The fish species identification system is a simple, fast, and accurate DNA-based method to identify the fish species present in a given sample. Fish samples are treated with proteinase K to release nucleic acids into solution. The fish genomic DNA is then isolated and PCR amplified using primers that bind to sequences found in all fish genomes. The PCR products are then digested with 3 different restriction enzymes and resolved on the Agilent 2100 bioanalyzer. The fragment lengths produced in the digestion reactions can be used to determine the species of fish, using restriction fragment length polymorphism, or RFLP, pattern matching software.
Hi, I'm Rachel Formosa from Agilent Technologies. Today we will show you a procedure for DNA-based fish species identification. This procedure is a screening method that allows you to identify the fish species present in either fresh, frozen, minced, cooked, dried, or otherwise processed samples, and this can be accomplished in less than 1 workday. We use this procedure to study seafood authenticity, which is very important to the seafood industry, as substitution and mislabeling can have significant economic, environmental, and food safety consequences. Plus, it can be especially difficult to determine fish species by visual identification and other traditional methods once a fish has been processed. DNA testing provides a much more accurate and reliable result. So, let's get started.
Begin preparing samples for DNA extraction by placing 10 to 1,000 milligrams of each raw or cooked fish tissue sample in 1.5 milliliter microcentrifuge tubes. To each fish sample, add 220 microliters of freshly prepared proteinase K solution and pipette up and down. Incubate the tubes at 65 degrees Celsius for 10 minutes.